Chapter 14, Section 13. Other electron acceptors. Gotta remember, we've been talking different, compounds. Excuse me, oxygen-based, sulfur-based, nitrogen-based that can accept that electron after the electron transport chain. Well, there are many others out there. Iron, molybdenum, or is that magnesium? Oh, I forget. What does that mean? Anyways, you can see these. These are common. So you can get, metal and metalloids, that can accept that final electron. A lot of this is going to be dependent upon the environment that microbe spent the past billion years, evolving and being selected for in. Some of them can, couple oxidation of hydrogen, acetate, and many other metals and minerals that are in the environment. And, that's what they pass on. Everything we've talked about now, up till now, have been cytoplasmic and extracellular, just on the other side of the cytoplasmic membrane in what would be a periplasm space for gram negatives, or just, before, in between the space, the cytoplasmic membrane and a peptidoglycan cell wall, or an S layer. Well, it turns out that some microbes extend this, metabolism further out away from the cell, and, based on what could be attached to either the cell itself or some of the cell appendages like flagella, pili, fimbria, and whatnot. Geobacter, the example on the previous slide, takes acetate, breaks it down, releases it as CO2 as part of its metabolism. Remember, acetate is a 2-carbon thing. Breaks it down, releases each of the carbons as, CO2. In the process, it's releasing electrons that are flowing down, interacting with, these pili. These pili that are conductive, that aid in retaining these electrons and just allowing them to spread down. And in the process, they're interacting with different iron-containing minerals. And then there's going to be different types of, heme or type cytochrome proteins. That's what these little red things are here. So you don't see the electrons as they're flowing down, but the cytochromes are interacting with them, and all of them are interacting with the iron-containing minerals, pulling out iron, pulling out, other electrons. We can have numerous other inorganics that are acting as the final electron acceptors. I mean, look at these: selenium, tellurium, arsenic, oxidized chlorine. These would kill us in seconds. The arsenic and the chlorines, definitely. But, microbes such as D-sulfotomaculum, and I'm not even going to try to do this pigmentum one, Aureopigmentium, can reduce arsenate, ASO4 to sulfate, orpiment, As2S3. So in the process of taking the arsenate and turning it into the orpiment, it detoxifies arsenic. Cool. This is Mother Nature's very own. Okay, we couldn't do this. We couldn't even figure out how to make this happen. But we do know that, this is great in biomineralization, taking arsenate that may be in the environment, may be in a toxic form, and turning it into a non-toxic sulfur mineral arsenic/arsenic compound. We're looking at these now when it comes to remediation. Superfund sites. Hey, look, whatever they're doing, lots of arsenic in the soil, it's killing everything. Well, spread microbes like this around. It's going to take a while, but we'll start to see the arsenic there being turned into something that is non-toxic. It's a mineral that will be trapped in the soils. And you can look here. You know, this bottle, of our desulfonectibulum in an arsenic-containing environment that can grow and grow very well. And in the process, producing that As2S3, which comes across as a, slightly pigmented or colored orpiment compound. Sorry, couldn't think of the word compound. We see that the anaerobes have spent, over a billion years coming up with alternate mechanisms. Because you got to remember, some of these, their lineages predate, almost all of them, their lineage predates oxygen environment. So they've had alternate mechanisms that some of the bacteria have retained, even though it's now in an oxygen, and we're in a very nice oxygenated atmosphere. They're growing in environments that are depleted or lack or whatever oxygen. E. coli can grow anaerobically on fumarate containing media. Turns out it uses the fumarate, reducing it to succinate, which is the opposite of the citric acid cycle. Citric acetate will take succinate, turns into fumarate, malate, oxaloacetate. So we do the reverse of it, and in the process, taking, that electron and a hydrogen, adding it to fumarate to get succinate. Citric acid cycle, if you don't remember, takes the succinate, turns it into fumarate, and pulls off a hydrogen / excited electron to reduce NAD to NADH. DMSO, dimethyl sulfoxide, trimethylamine oxide, TMAO. These are also two different compounds that can be reduced and accept, the oxygen here will accept the hydrogen and the now depleted electron at the end of the electron transport chain. So it's technically not using gaseous oxygen, it's using oxygen off of, an inorganic compound to form metabolic water, and in the process taking that depleted electron. And then there's also halogenated organic compounds that can come in and also be used. Benzoate, hydrochloric acid, chlorobenzenate will be reduced to benzenate and hydrochloric acid by certain microbes.